So let me switch over to the computer now, and we'll pick up with this slide. This is where we had left off. Those of you that will visit the test one again for a second time will know better now what neurophysin is after the discussion in the previous lecture where we had talked about the post-translational processing of oxytocin and vasopressin. I wanted to proceed to talk about the post-translational processing of proopiomelanocortin to reemphasize some of the key features that are common for these types of post-translational processing for hormones, going from a pre-pro hormone to a pro hormone, and then even further cleavage in order to be modified additionally. We saw that not only did we have cleavage as an example of post-translational processing for vasopressin and oxytocin, there was also disulfide bond linkage. There was glycosylation, so there were conformational changes due to the disulfide linkages. So we've seen 3 examples of post-translational processing so far as the examples. And the neurophycins ended up being just a carrier molecule to help deliver the hormones to the posterior pituitary to be released after the final cleavage as biologically active molecules. That makes sense to us. We don't want to make it a fully biologically active molecule, which remember is the most, not sensitive, but fill in the blank there. The most susceptible, thank you, brain, the most susceptible to the final enzyme cleavages that would destroy it so it's no longer biologically active. So there are post-translational processes that can occur, and we'll see some additional ones beyond the few that we listed so far with this POMC to further emphasize that example. So we try to keep it not in the biologically active form as long as we can until it's finally released and on its way to produce its biological effect. There are some other final post-translational processings procedures that are mechanisms that increase the half-life of the biologically active form of the molecule once it is released as well. And we'll see that with this example of the POMC. What will not be unfamiliar to all of you is, of course, my emphasis on using our imagination to envision the gene expression, cell-specific gene expression, tissue-specific gene expression, organ-specific, whatever it may be. We now are obviously in either a corticotrope or a melanotrope because we see the MSH being synthesized here as well. There's alpha and gamma MSH that can be produced, and beta MSH. Look at that. We got all kinds of MSH here. So these would be intermediate lobe melanotropes or pars distalis corticotropes that would contain, that would express this POMC gene. But now we're looking at enzymes that must somehow be involved. So it must be a cluster of enzymes, not just one enzyme, not just one endopeptidase that says, oh, I'm going to cleave here, 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 and here, and then in this cell we're going to end up with all of these hormones. This isn't always the case. There's differential gene expression of a population of cleavage enzymes in one cell such that ACTH is the predominant hormone produced in the corticotropes, and that MSH is produced in the melanotropes, and then at other times beta-lipotropin will be broken down. We know that if we exercise a lot, then our pituitary, the exercise high that they talk about, or if we're in extreme pain, those types of things, childbirth, beta endorphin is released from the pituitary gland to help relieve the pain, producing the runner's high and that kind of stuff. So what happens in the corticotrope such that now all of a sudden the beta-lipotropin is going to be cleaved to the beta endorphin so that beta endorphin is now released from basically the same corticotrope that would be releasing the ACTH. At the same time we're experiencing pain and runner's high, the ACTH is going to be released as well. Maybe in the runner's high, ACTH is released because of the low blood glucose, you think? Another question on the first exam. So now that I've set the stage for this, let's read through this statement on the biosynthesis of ACTH. So prohormone convertase enzymes, PC1 and PC2, process POMC in Figure 1. So there's at least two types of hormones that are responsible for those initial cleavages of the prohormone to the some of the byproducts, at least ACTH being one of the hormones as a byproduct of that first cleavage. And we'll look at it in more detail in this illustration as well. But after that first cleavage, we see that ACTH is one of the hormones that is biologically active byproducts that is produced. The beta LPH does not have significant biological activity that I'm aware of, but certainly the ACTH does, and this N-terminal glycopeptide, as far as I'm aware, does not have a biological activity. So a couple of hormones already identified, enzymes identified. So this has been an intriguing challenge for scientists to find out how is this post-translational processing, these cleavages, regulated. It's very important to understand it, because if there's some way we can discover a drug to target selectively one process of it, we might be able to fix a clinical situation such that more ACTH is made to be available, or some drug to help alleviate pain that could shift and pump out the beta endorphin. So there's, other than just the curiosity, it drives me crazy, how does this get done? How does this happen biologically? There is that type of an interest in figuring this out as well. And we said it's always pairs of lyse-lyse or lyse-arg amino acid residues. So knowing the primary sequence of hormones and prohormones and pre-prohormones is important. So that first cleavage produces the ACTH, as we said, and the other molecules, the beta-lipotropin. So the beta-lipotropin must also be beta-lipotropic hormone. Otherwise, how do they get the LPH acronym? Anyway, some of this is above my head too, so I'm there with you. Don't feel alone. So the ACTH, as we said, as we noticed, can be processed further, generating the alpha-melanocyte stimulating hormone and this CLIP, the corticotropin-like intermediate lobe peptide, whereas the beta-LPH can be processed, as we saw, ultimately to provide the beta-LPH and the beta-endorphin. Should be beta-LPH to delta-LPH. I knew that was a mistake on that slide. This beta here should be delta, a byproduct. The amino acid sequence, the number of residues is decreased because it has been cleaved to provide the beta-endorphin molecule. So in the corticotropes, so now we're distinguishing the pars distalis from the pars intermedia, where the alpha MSH should not be produced. It's POMC is processed to the N-terminal glycopeptide, joining peptide, the ACTH and the beta-lipotropin, as we had stated, as we had surmised just from looking at the possibilities. We want it to stop there in the corticotropes so that we can have ACTH to release. Then if necessary later, the same cell can, under whatever stimulus, can cleave this and release the beta-endorphin. It's not perfect, though. Nothing in biology is perfect. So anytime we have an enzymatic cascade, whether it's degradation, cleavage, or just hydroxylation or anything like that, decarboxylation, we always have intermediates, and there's always one form of one of the molecules that accumulates to significant levels, and typically is the one that is released predominantly but everything else can be detected, either upstream of that major molecule like the ACTH or even downstream. Just somehow the cell gauges everything so that norepinephrine is what I'm going to release, but some of my dopamine is going to escape. And as well, in this case, the anterior pituitary cells are going to secrete, the corticotropes secrete mostly of the pars distalis, mostly the ACTH, but if you could capture one of those cells, you'd pick up a little bit of alpha MSH, and so on and so forth. There are other post-translational modifications, glycosylation, N-terminal glycopeptide addition, as we had said. That's the glycosylation. And C-terminal amidation of the N-terminal glycopeptide, the joining peptide, and the alpha MSH, and N-terminal acetylation of the alpha MSH and beta endorphin. So depending upon the cleavage byproducts that are produced, there's additional post-translational processing that can occur to those, and that, of course, is summarized in this slide. So the N-terminal acetylation is shown as the little red hemicircle on the left. It's kind of a hemicircle, and that only happens with this molecule up here. That's the alpha MSH produced by the cleavage of the ACTH. And as well, C-terminal amidation. This will be pretty common, as we'll see as we proceed to the semester. This post-translational processing of the C-terminal amidation on the joining peptide as well as the alpha MSH. We see beta endorphin also gets the N-terminal acetylation. This amidation as well as the acetylation both contribute to an extended half-life for the molecule. The smaller the peptide, we would predict the shorter the half-life. If you, the two that I used in the general physiology course on the exam they had was thyrotropin-releasing hormone, that's a tripeptide, and insulin, that's definitely a much larger molecule than that, and asked which would they predict to have the shorter half-life. We would expect that a tripeptide, 3 amino acid residues, one of those gets cleaved. There's only 3, so cleave it. It's got to be one of them that comes off, 1 and 2. Then you no longer have a biologically active molecule. So it should be very easy to remove half of the critical amount released or contributed to the body fluid to reduce the half-life. And I have a link to a site, a couple sites that I shared with them after I asked them questions about half-life, and they weren't really happy with my responses. So I'll pass those on to you so you can go and take a look at half-life questions. I mean, get more assistance with understanding half-life. And obviously I'm planning, subconsciously slipped out there, to have a question or two on exam 2, test 2, relative to the half-life of molecules in the blood. Didn't suppress that one very well at all, did we? So we now have glycosylation, cleavage, conformational changes such as disulfide bond linkages, N-terminal acetylation, C-terminal amidation, and the glycosylation is shown here. It's kind of strange that these inactive portions are the ones that we think are biologically inactive and maybe insignificant, are the ones that are getting glycosylated. It makes you curious about why that would be occurring. I really don't know, and very likely no one knows exactly what's going on there. So now let's make the transition. I know we're talking about the synthesis still of hormones, and we're to proceed to basically the biological activity of them, because we're talking about post-translational processing to make them biologically active. Now the next step should be, what's the biological action? And of course we'll talk about receptors with the neurotransmitters, so I'm not going to spend time on that now. But then we should transition to the degradation, the removal. So we've got to synthesize, secrete, or make it biologically, biologically active, secrete it, let it have its biological effect, and then degrade it to remove it, so that it is a transient signal. It's not just present all the time, so we never know that anything changed. We have a baseline, either we increase or decrease with reference to that baseline. But I do want to take a moment, because this information was available from the Endotext source, I think it was, or it might be the ACNP site, on the POMC promoter. So if we're thinking about this POMC protein that can be cleaved into a variety of biologically active hormones, let's go back to the level of the gene. Like, as you know, I always like to try to do that. So the promoter region is very important for helping to understand what is going on. We can make some predictions. Going to be a TATA box, a CAT box. They're almost always there. But what is the AP1 site? And what are some of these other things that are mentioned? I present them at this time because we will probably never talk about the POMC gene again. But we will, as we proceed to talking in greater detail about each of the neuroendocrine axes, focus on the information that is presently known about the promoter region for each of the genes, whether it's the hypothalamic neurohormone, or its receptor in the anterior pituitary, or the hormone to be synthesized and secreted in the anterior pituitary, and then on down the line. So here we are, the POMC promoter region in the anterior pituitary. So TATA box and CAT box, got to have the polymerase, the RNA polymerase 2, the site for the binding of the RNA polymerase, and these are involved in that. And then everything else is involved in making it more or less available for the polymerase to bind, as we saw in a previous illustration on a slide in the earlier lecture. So AP1 is a response element on promoter regions. At the moment, I can't recall exactly what the AP stands for, but this is an acronym for a chemical that, when added, would change gene expression. And they kept chasing down the genes and then get promoter regions. So first they'd see protein levels would change when they'd add this chemical. Then they would get the gene for that protein, find the messenger RNA, then the gene. Let's get backwards here in sequence. And then look at the promoter region. And then when they started comparing the sequence of the promoter regions, there would always be this sequence that they'd find in every one of them. And as they began to mutate all those and to study them, if they mutated that portion, then this AP molecule would no longer have an effect. So they called it the AP consensus sequence. So it's a sequence that always showed up, so it was a consensus. It was always there. When it was there, there's a consensus basically that you add this AP1, this AP chemical, and you're going to change the expression of that gene downstream of that promoter. And there's obviously more than one of them because there's the AP1. We'll see that a family of proteins called Foss proteins that were discovered from feline osteosarcoma cells. So that's the FOS, and the C was the canine. No. I don't know what the C was. Anyway, the CFOS is the acronym for it now, and everybody recognizes it, but stop asking what all the letters stand for. And there's JUN, June. So there's CFOS and June that are these strange acronyms that molecular biologists come up with for these transcription factor proteins that we'll talk about later when we begin talking about intracellular signaling. But the Foss and the June are the transcription factors that bind to this consensus sequence. So it became very important because it was associated with osteosarcoma and was related to changing gene expression, but I think it turned out it didn't really have much to do with causing cancers. It was just a tool that was utilized by some other gene that was the real troublemaker. Anyway, this is how we discover things and becomes useful. It's a very important response element. So a consensus sequence can become a response element once you find the transcription factor that binds to it and produces the response. Then we call it a regulatory element. RE is the abbreviation that we talked about. That regulatory element is a sequence of DNA in the promoter region. Transcription factor binds to alter the transcription rate for the gene attached to that promoter region, downstream to that promoter region. So I know it's a lot of words, but it's recorded. You can back it up and play it again and again, and it's the same story every time. And we saw it with my illustration of it. It was in the introductory supplementary lectures about promoter regions and these regulatory elements, these REs. So AP1 is one type of regulatory element that can also be called a response element because you add a chemical like AP and the gene responds. So this is the element that responds to it, so it's a response element. But first you have to change all that DNA on the promoter region to find out which portion is responsible for this AP producing a response. When you find it and it shows up on every gene that responds to this AP chemical, then you call it a consensus sequence. It always shows up. So I'm going to be using the term consensus sequence, response element, or regulatory element, and the two latter ones are the same, abbreviated to RE. So the promoter has those. So now once you know the sequence of the AP1 consensus sequence, when you sequence a promoter region for another gene, AP1 site might show up. And nobody ever tested whether this AP molecule or the C-Foss and C-Jun molecules have anything to do with regulating that gene. But if that consensus sequence shows up, you assume that you have a response element. You then have to go on to further test to see if indeed it does. So I don't know if anyone has tested whether the C-Foss and C-Jun have any functional effect on this AP1 consensus sequence candidate response element on the promoter region of POMC, but from sequencing it shows up. The corticotrope and the melanotrope specific transcription of POMC appears to be dependent on a consensus sequence, right? So you start making mutations in the promoter region and see if the corticotropes and the melanotropes at a cell-specific level continue to express that gene. So the CANN, this just means that it could be any nucleotide. TG is the element motif. It is the consensus sequence. So we don't know what transcription factor might be binding to it necessarily. It's all conjecture from molecular test that it is responsible for the cell-specific expression until we've tested if it's a true response element and demonstrate it's a regulatory element by first finding the protein, the transcription factor that binds to it. Then we could call it a regulatory element. But until then, it's just an element motif that is considered to be essential because if you remove it or mutate it, you no longer get the cell-specific expression. It looks like they've got synergistically binding corticotrope upstream transcription element binding proteins, the cute proteins. So it looks like somebody got the protein on this little cutie here. Also note that the A is complementary to the T, and the G is complementary to the C, right? I've got to remember the term for it. Okay, I'll give you the term for that. But they're short sequences that have tremendous effects, as we see. Just 6 nucleic acid residues, and 2 of them don't even have to be the same thing all the time. They just have to be in this sequence. Come on, brain. Okay, it's not going to pull out the term for that. I'll have to come at another time. So the protein has been identified. So they're probably at one step closer to identifying it as the true cell-specific regulatory regulatory element for the corticotrope and melanotrope to express the POMC gene. There are other, this is a collection of proteins. These cute proteins include NeuroD, PTX, and TPIT. So the PIT, I think, is one that we had recognized before as being related to cell-specific gene expression, actually tissue-specific gene expression in the anterior pituitary. I don't know any details on any of these. I haven't done research on them or literature research. Over the years, as I see new bits of information, I try to find a paper here and there and then use that paper as an example to build upon these. So we can't know everything, but we try to pick it apart to understand what it is from the context. So then the POMC promoter, continuing, the transcription is positively regulated by corticotropin-releasing hormone, CRH, vita infra. Hopefully somebody's had some Latin and they can understand what that means. By inference, I don't know. Obviously, corticotropin-releasing hormone should increase the expression of the POMC gene. In other words, POMC transcription is positively regulated. It should increase gene expression when CRH binds to the cognate receptor on the corticotrope or melanotrope. So it acts via a G protein coupled receptor, activating adenylate cyclase, increasing cyclic AMP, which is very common for almost all of the hypothalamic hormones regulating anterior pituitary cell secretion. So that's something that is conserved across those. Also stimulating protein kinase A. We'll go into details on all of these at a later time. Protein kinase A phosphorylates a transcription factor called cyclic AMP response element, binding protein. I think that was a CREBP, wasn't it? CREB or a CREB. And in this case, the transcription stimulation is mediated by an upstream element. The CRE we see embedded within here, cyclic AMP response element, but it appears that they've added to it P, pituitary corticotropin releasing hormone response element, binding a novel transcription factor, PCRH-REB, containing protein kinase A phosphorylation sites. So I'll have to investigate this more myself to go to this reference from the source to see. This might be a brand new protein that is specific for corticotropin releasing hormone, not the CREB that we will study in more detail in class. So there's more and more information being discovered all the time about this, especially for the hypothalamic pituitary adrenal axis, because it is so clinically relevant. There are many disruptions or pathologies associated with disruption of the HPA axis. So the CRH also stimulates the transcription of CFOS, FOSB, JUNB. So here are these proteins that I said that interact to bind to that AP1 site. The pituitary adenylate cyclase peptide, so PACAP, also stimulates cyclic AMP synthesis and POMC transcription, presumably through a common pathway with CRH. So I know all this doesn't make much sense right now, but what I'm promising you is that by the time we push on through the semester and struggle with it all, most of this will make sense just intuitively as we continue to talk about promoter regions and build on our knowledge of consensus sequences, response elements, regulatory elements, and the various types and classes of transcription factors. We'll build a safe zone with a small number of them and then be able to say, oh, gee, there's a transcription factor they're describing. I don't know anything about it, but that's okay. I got my comfort zone, and I'm going to be all right. And just develop our respect for the wealth of knowledge that is growing and growing so rapidly all the time as people chase down these experiments. Then what we'll see is that there's a lot known about this axis, a lot known about that axis, especially HPA and HPG, the stress, clinically relevant one, and reproduction. Then you get to some of the others, and we don't know a lot about them yet. Why? Not a lot of pathologies, not a lot of human patients, not a lot of money to research them, so we just don't have the knowledge. Might be the people that are interested, but there isn't a large amount of money to support that type of research. So we can see the impact just in the literature and our knowledge base from that social quirk there. The mRNA transcription is negatively regulated by glucocorticoids. Intuitively, oh yeah, negative feedback should decrease synthesis and secretion of ACTH. So I would predict that the POMC promoter does have a glucocorticoid response element, or GRE, that the glucocorticoid receptor would bind to. So the effect appears in the rat POMC promoter to be dependent on a negative glucocorticoid response element, partially overlapping the cat box. So interfering with the binding of the RNA polymerase. The element binds the glucocorticoid receptor once it is ligated by glucocorticoid as a homodimer plus a monomer on the other side of the DNA helix. So that's something new. I'll have to dig into that and add to what we discuss. So we're going to develop the skills and the knowledge to understand what is being stated here. We talked about CFOS and the June. There's CFOS, FOSB, C-June, June B, all these different transcription factors. And when the cell is activated, two of them are going to come together and form a dimer. If you get Foss and June together, they're not the same. That's a heterodimer. The glucocorticoid receptor has to be ligated, and then it'll stick together. That's a homodimer. So di, two, mer, two molecules. Do we all remember some of our protein chemistry there, hopefully? And they're different, so they're heterodimers. So all that's going to come into play, and we'll just know automatically. The AP1 site response element requires a heterodimer of Foss June. It can be C-Foss and C-June, or C-Foss and B-June, or Foss-B, and whatever else. They have to be phosphorylated before they form the heterodimer. Then they combine. So it'll go on and on and on. But we should become fluent in describing these types of events by the end of the semester. That is our goal. So we're building up to it, just predicting what we'll see in the future. You don't need to know all of this. Just start to build on the intuitiveness. POMC gene its expression should be increased when CRH binds to its receptor on the corticotrope. We just talked about that. When glucocorticoid levels increase in the plasma, then negative feedback should kick in and decrease synthesis and release. And basically what it does is it decreases the amount of synthesis of the readily releasable pool, so there's less to be released. As well, it's going to remove the stimulus, it's going to inhibit the CRH. So we'll build on this, which should become more and more intuitive as we proceed through the semester. So as I place greater and greater expectations on your intuition from test to test, expect more challenging questions, right? And more detailed information. I don't make it impossible. If I trip you up, I come back to save you. I want you to learn and do well in the course, so don't go running off just because you didn't do well on a test yet, okay? I'm very nurturing in that sense, so send me an email and we'll work it out. So the glucocorticoid, let's see, where were we? Yeah, the homodimer is required to bind to the DNA. The regulation of transcription may also be indirectly mediated via downregulation of C-June expression and direct protein-protein mediated inhibition of CRH-induced AP1 binding, as well as inhibition of CRH receptor transcription. Oh, and then there's a reference number 18 there. So this is all obviously on the printout that you can have. So it obviously becomes more complex. Not only is the glucocorticoid going to produce negative feedback by interfering with the cat box by acting as a transcription factor, inhibiting transcription in this case, I lost my arrow setting here. It's also going to interfere with other regulatory elements and the binding of their transcription factors, i.e. AP1, the C-Foss, C-Jun combination, heterodimer, that drives the expression, increases expression. So this glucocorticoid response element, when bound by the ligated homodimer of the glucocorticoid receptor, is going to say, okay, I'm going to mess with the cat box and directly make it more difficult for the RNA polymerase to bind, so reducing transcription that way. But I'm also going to mess with the AP1 site so that the gas pedal coming for increasing gene expression coming from the CRH, the drive, is less effective. So in both ways, I'm suppressing the expression of this gene. RNA polymerase isn't going to bind as much, and whatever, most likely kind of loosening it up to make the cat and tata box more available that the AP1 site was doing. I'm going to mess with that and reverse that as well. So I'm going to reduce gene expression through two mechanisms. And this is new since I taught the course, I think the last time was about 1997. So this is new information, and it's very interesting. So at those times we questioned how might the GRE be acting to reduce gene expression in this case. Now more information is available, and we see it here. So leukemia inhibitory factor, a pro-inflammatory cytokine expressed by corticotropes. So one last chance to toss in that neuroendocrinology or neuroendocrine immunology, all those terms that we talked about very early on. One last chance to toss in some of that. It is a pro-inflammatory cytokine expressed in corticotropes, has also been shown to stimulate POMC transcription by activation of JAK-STAT pathway. I think this is part of a cellular stress response pathway. This stimulation is synergistic with CRH, so they act together to produce a greater increase in the gene expression. Deletional analysis of the POMC promoter has identified a LIF, responsive region. So they're looking for the consensus sequence, the response element, from negative 407 to negative 301. We're going to hit a point when we talk about methods. We're going to talk about how this is done. We'll illustrate it with drawings on the overhead projector here. And we'll, again, become fluent and conversant in how this is done. We won't become technical experts, but we will gain enough knowledge about the methodological approach to appreciate the strategy. What are they doing? Obviously, they get the promoter region for POMC and sequence it, and then somebody remakes it, but they don't put these couple of guys in, and then they use it to test to see if it's functional and responds to LIF. If they delete or mutate, or what's the other one where you replace it? Forgot the term for that. Replace it with another nucleic acid, and suddenly it's no longer responsive when you add the LIF. Then, aha, I not only have a consensus sequence, I have a response element. Now let's test the whole gene and see if it's involved in regulating this gene under normal biological conditions. Indeed, my experiments demonstrate that. I now have a regulatory element. So we'll build on that technical knowledge when we cover the methods. So it's thought that this pathway might form an interface between the immune system and regulation of the HPA, which is what we had talked about all along. Now then, we want to talk about degradation, and we take from one of our sites, either the ACNP, I think is the source of this site, a textbook on psychopharmacology, and talk about degradation and biological half-life of Galanin. It was one of the the peptides on the list at the very beginning of this lecture. So the agonist action of Galanin and its N-terminal fragments is terminated by their degradation at the N-terminus. So an exopeptidase by enzymes which cleave the first or the first two residues. So it's kind of an exo-endo peptidase. So it's an N-terminal exopeptidase would be my recall of my biochemistry to name this. With GW, it's got me here, I don't know what that is, yielding the inactive 3 through 29 of 30 and 3 through 16 fragments. So by just removing a couple of amino acid residues, we've inactivated this protein, this hormone, Galanin, or this neuromodulator, as studied in the spinal cord and hypothalamus. So they probably took hypothalamic and spinal cord tissue, homogenated it, added some of this protein peptide exogenously, and then, after periods of time, looked for it in alternate forms and identified where it was cleaved, and therefore, in the interstitial fluid or present in the tissue, the central nervous system, the degradative enzymes exist to destroy these biologically active neuropeptides, which is what we predicted and what we would expect. So the data suggests that these diaminoacyl peptidases, which cleave and inactivate Galanin, may belong to the class of metalloproteases because chelating agents afford some inhibition of the degradation of Galanin by cerebrospinal fluid and by spinal cord membranes. So they might be degradative enzymes exposed on the outside of neurons or other neural tissue that are just there to destroy peptides present in the interstitial fluid. So we're beginning to build this picture in our mind of the mechanism of degradation of the peptides, hormones, and neuropeptides. So now we introduce this term, the biological half-life of Galanin upon incubation with rat CSF or with hypothalamic membranes is 60 minutes. That's actually quite long. So it's about 10-fold longer than that of equal-sized peptide, the VIP that we mentioned before, vasoactive intestinal peptide. So each neuropeptide, each hormone is going to have its own half-life. The time period that it takes for a stable concentration of the molecule, typically in the blood, but in this case in their solution where they put the cerebrospinal fluid and then added it in, they had a final concentration, and then you measure the time it takes for that concentration to reach 1/2 that level. That's the half-life. If it's plasma, you inject somebody with something, or the hypothalamus pumps more ADH into the blood, it distributes, reaches a new level. How long does it take for that to reach 1/2 that concentration is the half-life. And as I said, I'll send you links to these other sites where you can investigate that more. One, you can actually put numbers in, and it'll calculate the half-life for you. So this is pretty high, 60 minutes for a peptide. I don't know how many amino acids this is, but even if it was 40 amino acid residues, 60 minutes is a long time period, a long half-life. So in 1 hour, it's reached 1/2 of the concentration. So the long half-life may enable Galanin to exert paracrine action. So we know what they're talking about, acting on something else. So the next stage, related even to the textbook, is to compare the neuropeptides to neurotransmission of classical neurotransmitters. And what I'm going to do to stay on schedule is I'll record this on my own computer, so you won't see me in this little picture-in-a-picture, and I'll post it, and it'll be available for you to view. We'll talk about this again when we talk about the neurotransmitters, and there's no harm in that. What I'm going to do is flip through that real quick and mention the other topics that are described in Chapter 11, and to allow us not to be concerned. Importance of neuropeptide agonists and antagonists, no problem. We'll talk about that when we talk about specific papers. Identification and localization of neuropeptide target cells in the brain. We'll talk about this when we talk about methods and get into specifics. And then the neuropeptide activation of second messenger systems. This will come certainly in the lectures on receptor-mediated mechanisms. Man, they've got me in the pip. I'm totally out of focus. You can see downtown, yeah. So don't be concerned about these in this chapter. We'll get to it at another time. So I'm going to stop here and let you know that I'll complete this series of slides in a separate portion that will be present on the WebCT course site, and the lecture is already there in PDF, not in this blue and yellow, but in black and white, so it should print very nicely. Appreciate your time and attention. For those of you in the time today that this is recorded, thank you for coming. 2 people. Did you have a question? Yes. Did you want to ask something? Yeah.
Oh, no, I'm fine. Thank you.
Okay. Bye-bye.
Bye.
You're welcome. Have a good weekend.